Hello, this is Dr. McIntyr from Washington State University Puyallup Research and Extension Center. I'm going to talk to you today about stormwater runoff, toxicity, and treatment. So the reason stormwater runoff is a problem in urban areas is because this is not a forest. There's nowhere for runoff to go when it rains, and it picks up contaminants and brings them usually to the nearest water body. This video shows what that looks like in Puget Sound, where a stormwater outfall enters the water off of Alki Beach in Seattle. So this is obviously the part of the stormwater pollution that you see. You see the sediments, you see the turbidity. What you don't see are all of the chemicals that are also carried in that runoff, and that's what I'll be talking about today. Now, the reason we care about this is because of the aquatic wildlife that resides in our streams, that relies on our water bodies to survive. Besides the importance to the ecosystem of these animals, they're also important regionally, in particular salmon, culturally and economically. So we talk about Coho salmon in particular as being a sentinel for stormwater impacts. In the Puget Sound region, which you see on this map, all those little blue squiggly lines are the tributaries and the river systems where Coho salmon reside either currently or historically. So they're very widely distributed, and they do prefer lowland streams, which of course is where humans like to build their homes as well. So there's a lot of potential for overlap there. Coho salmon also spend an entire year in fresh water during their early life, which is longer than most of the other Pacific salmon and trout species. So that's fully a half of their lifetime spent in fresh water, and this is where we're going to have the most potential for stormwater impacts on this species. I'm going to talk about two examples from the Coho life history where we have seen integrated impacts of stormwater runoff in the field. The first is adult spawners returning to creeks to lay their eggs in the fall. What we've seen is that throughout the urbanized environment in Puget Sound, we see pre-spawn mortality. So these adult Coho salmon have come back to spawn, and they're dying before they get a chance to spawn. Anecdotally, we see this all the way up into southern British Columbia and all the way down into northern California. Very high rates of pre-spawn mortality. And what this looks like before they die is a series of symptoms that are very particular. So they include things like disorientation, they include lethargy, they include a loss of equilibrium, and then finally fin splaying. We also see surface swimming and gaping, like in the top right video right now. So these are not healthy fish, and they're obviously being affected by something in the water, and it does seem to be tied to the timing of storm events. So the second example that I'm going to show is from Coho salmon embryo development. So out at Longfellow Creek in Seattle, we have somewhat of an outdoor laboratory. And there we have done an experimental filtration of the urban creek water. So in the images you see here, the top right one is the regular urban creek water. Sometimes it's clearer, sometimes it's murkier, but in comparison, we have water that we have tried experimentally to filter. So we've used a sand filter, charcoal and activated carbon, just to compare what a somewhat filtered water would be like compared to the unfiltered creek water in terms of Coho embryo development. So we raised embryos in these waters, and these are straight paths out of the creek, filtered or not filtered, passed over the embryos, returned to the creek, and new water continuously coming in. What we saw is that in the filtered stream water, Coho embryos developed fairly normally. We had about 10% of the embryos that were either abnormal in some way or had died at about this 50 days of development period. In contrast, the coho embryos developing in the unfiltered stream water, this is just the regular urban stream water, was quite a different story. 75% of those embryos were either abnormal in some way or had died at this point, by this point of development. This arrow is pointing to cranial hemorrhaging, so actual blood spots accumulated in the head of embryos. This image shows just a top-down view of what that looks like. In the filtered water, we saw what we expected, which is this symmetrical pattern of vasculature in the head region of the fish. And in the unfiltered stream water, we don't see that. Instead, we just see some random blood spots. So those are integrated examples of real-world impacts of stormwater runoff. So a few years ago, we started a program specifically to get more into the toxicity of urban runoff. What's actually in it chemically, characterize it chemically, how toxic is it, and could we prevent it using something like a low-impact development technology? The runoff that we started to use was collected off of an urban highway. And the reason for this is that it contains it's a concentrated source of a lot of the chemicals that we're concerned about in the urban environment across different land uses. Also, we chose this source because we could collect easily off of this elevated highway from downspouts that were located in the parking lot of our facility. So, capturing the runoff, we did thoroughly test it chemically, which I'm not going to talk about today. That's you can find that in some of our published papers that I'll show a slide for a little bit later on. But what we see across different storm events are a suite of sublethal effects in developing fish embryos. Now for this we're using zebrafish, which is a very popular aquatic lab rat type animal. We know a lot about them physiologically, and toxicologically they actually are very responsive to chemicals in much the way that humans are. So there's a lot of human medical research that's based on this animal model. Now some of the stormwater runoff events were actually lethal to the zebrafish embryos, but we were more concerned with these sublethal effects because that's what we expect to see out in the environment where runoff like this is diluted. All right, so some of the effects that we see include an inability to hatch or a delay in hatching. We see developmental delays, so smaller embryos, just like in that coho example. A small eye phenotype, this is called microphthalmia. And pericardial edema, that's where the yellow arrow is pointing at the region where the heart is developing. And in that area, we'll see fluid accumulation. And this usually means that there's a problem with the developing heart. In fact, this is a symptom that you also see in humans with congestive heart failure, is this edema. And finally, in more extreme cases, you see this severe pericardial edema. You actually can see a deformed heart and other craniofacial deformities. I have here some videos of what this looks like. So the top videos are from zebrafish that are 48 hours old, and the bottom images are different only in that the fish are now 96 hours old. Now, these are different fish exposed to different runoff treatments, but in general, what we're seeing here on the left with the controls is a normally beating heart. You can't necessarily see, but the fish heart has two chambers, and you can see the blood pumping from one chamber to the next. In the fish exposed to the runoff, we see a variety of these sublethal defects from exposure to the urban runoff. So they include the pericardial edema, that's swelling around the heart. They include blood pooling behind the heart because it's not able to be pumped into the heart and through the body at the correct rate. We see this cranial hemorrhaging, just like in the coho embryos. And we'll also see a deformed heart. Here you can see that the heart does have two chambers, but in fact, for a heart to function properly in this species, the heart has to then loop It has to be folded in a particular manner, and if that doesn't happen, which chemicals can interfere with, then a lot of the other developmental sequences get interfered with. So one of the things we wanted to do was look at this pre-spawn mortality in terms of urban runoff more directly instead of the more speculative approach we had to take in the past. So we wanted to see, could we cause pre-spawn mortality in otherwise healthy coho by exposing them to urban runoff? So we worked with the Suquamish tribe out at their Grover's Creek facility near Poulsbo, where the fish enter through Miller Bay up a relatively short freshwater section into the pond where they are collected by the tribe. And this is very similar to the situation at Longfellow Creek, where we had studied for many years pre-spawn mortality. We collected runoff in Seattle from the elevated urban highway, and we brought it out to the hatchery where we exposed adult coho to either clean well water or to the urban stormwater runoff. Each of these tubes contains one adult coho salmon, and they have water and air flowing over their gills so that we can maintain them in these exposures without any trouble. After 3.5 or 4 hours, we take the fish out of those tubes and put them into clean water for observation. Here on the left you see an adult salmon that's been in the control exposure for 3.5 hours, and it's behaving as we would expect, a little disoriented, having been released from a tube into this observation chamber, but otherwise very active and responsive, and a little bit aggressive actually. In contrast, the adult coho that was exposed to stormwater runoff for the same amount of time is showing some advanced symptoms that look very much like those of the pre-spawn mortality we see in the field. Loss of equilibrium and an inability to swim at this point. So this is followed very shortly after by death. Now, what we've seen with our science studying this, what is the problem of stormwater runoff? What's the nature of the problem? And we've been moving towards what is the solution. So with that, enter green stormwater infrastructure, also called low-impact development. So this is things like green roofs, like pervious pavement, like rain gardens. And many of these technologies employ bioretention, which is a way of getting runoff to infiltrate into soils as a treatment method both for hydrology and contaminant removal. Now at Washington State University in Puyallup, we have a facility where we study some of these technologies, including permeable pavement, replicated plots, and all of these are replicated, which makes them very amenable to scientific study. We have rain gardens where we're testing different plant palettes, and then we have mesocosms, which do contain plants. They actually contain all the same plants, but we're testing the effect of different soils on removal of pollutants. Now the work I'm going to show you that we've been focusing on so far in terms of the biological effectiveness are these large soil columns. And they're sort of halfway between the field and the lab. I think of it as taking a big core section through a rain garden. So at the bottom of this column you have a drainage layer of gravel, and on top of that you have bioretention soil medium. So for our first experiment with bioretention, we again collected runoff from the urban highway, brought it to the greenhouse where these large soil columns are. And for this study we had collected them in glass carboys, so we had to homogenize the sample before we could begin testing. We mixed the sample in this large cistern and then drew off what would be the unfiltered stormwater runoff. The rest of the runoff then went through these large soil columns. Half of them had plants and the other half did not. And I do want to say too that the bioretention soil media used in this particular experiment was mostly sand, so it's 60% sand, 15% compost, 15% shredded bark, and then 10% of water treatment residuals. We've done very similar work with the 60-40 mixture. I'll describe it in a moment here. So we collected the effluent that came out of these columns and then brought them to our fish laboratory, where we tested them directly on our target organism of interest, juvenile coho salmon. So would the filtration prevent the toxicity that we would otherwise expect to see? Here we show the juvenile salmon in their exposure aquaria. We have the controls, we have the unfiltered runoff, what we're just calling runoff here, and then the filtered runoff, either with plants or without plants. Now what we saw is that the fish in the unfiltered runoff treatment, there was 100% mortality, and this happened actually fairly quickly, within 12 hours of exposure. Typically, this standard toxicity test is 96 hours, a full 4 days, to assess the differences in survival. But those fish had all died in far less than a day in the unfiltered runoff. In contrast, the fish in the control treatment, in just the regular fish laboratory water, had 100% survival and no illness. So what was going to happen in the filtered runoff when we take runoff that killed all of the fish in less than 12 hours and we simply filter it through a soil system? Well, actually, we saw 100% survival and no evidence of illness in these fish, and that was at the end of the full 96-hour period. We also saved water from these exposures and, oh shoot, here we go. We also saved water from these exposures and tested them on some other animals, including the zebrafish embryos, also including some invertebrates, wild mayfly nymphs, and a water flea, Cereodaphnia dubia. This shows the sublethal effects on the zebrafish embryos. And we have here the proportion of fish that were able to inflate their swim bladder, their air bladder, fish size, that pericardial area, the area around the heart. So larger area means more swelling around the heart, and then the eye area. And what we saw is that fish exposed to the unfiltered runoff, none of them had their air bladder inflated at the end of the test period. They had smaller eyes. They had enlarged pericardial area, so they had swelling around their hearts. And they also had significantly smaller eyes compared to controls. However, when we treated the runoff, we saw that that was completely reversed in most cases. So we now see normal air bladder inflation, normal size, normal hearts, and almost normal eyes. You can see that there's a huge improvement back towards control eye size, but there was a slight significant reduction compared to controls. Another thing that we see in the stormwater runoff is that we can look at the molecular level, and one of the things we can look for is exposure to contaminants, and we do that by looking at this enzyme, CYP1A, which is part of a detoxification pathway for aromatic hydrocarbons. Polycyclic aromatic hydrocarbons, for example, many of them trigger this detox pathway, and a variety of other aromatic hydrocarbon contaminants as well. So this green color is actually where that enzyme is located in the fish. And here we're seeing a surface photograph of the front half of an embryo exposed to urban stormwater runoff. So here's its large eye, and you can see the red up at top is it's actually just the muscle in its pectoral fin. So the first thing we note is that the skin of the fish is showing this induction or an increase in this detoxification enzyme. So obviously the fish's skin is being exposed to these aromatic hydrocarbon contaminants. Now if we zoom into the heart region of this fish with this type of confocal microscopy, we can actually go inside the fish and look and see what's going on there. So the red stain in this part of the picture is actually showing us where the heart muscle tissue is. Now the green on the outside, on the left, is the skin again. But the green inside the heart is showing us that there's detoxification enzyme being produced within the heart tissue itself. So we know that the heart is a direct target for some of the contaminants in the road runoff. And what we see when we filter the runoff is this difference. So let me describe what you're seeing here. On the left is some images from the control fish. It's hard to tell what you're looking at because you can't see the green outline of the skin of the fish, but it's a close-up of that heart region. You see the red as the heart muscle. There's no green staining because in the control fish, there's not really a need for this detox enzyme. And the bottom image is actually the tail section, so showing you can see the muscles in the trunk there. In the middle image for the runoff, we see that same picture of the heart with the SIP induction in the skin and also in the endocardial tissues, that's within the heart. In the bottom picture with the tail, we see green throughout the tail region in the vasculature. So we know also that the SIP enzyme is being requested for detoxification duties in the peripheral vasculature as well, not just inside the heart. And the final picture shows fish that have been exposed just to runoff that went through that bioretention system. And what we see is a complete loss of the SIP induction in the skin of the fish, and also a complete loss of the induction of this enzyme inside the heart. Now you can see by the arrow at the top that there's a little bit of peripheral induction in the vasculature, in the peripheral vasculature, of the zebrafish. And the bottom picture there shows that there's still a little bit in the peripheral vasculature of the trunk. But very importantly, there's none inside the heart anymore. And so this agrees with what we see in terms of a loss of edema, for example, swelling around the heart of these fish after the runoff has been treated with bioretention. This table is summarizing all of the different tests that we did to look at effectiveness in terms of biology for treating runoff with bioretention. So different organisms, different endpoints, different time points, and we see that almost all of those signs of toxicity have been completely eliminated by the bioretention filtration. Now, microphthalmia, we saw there was still a tiny bit, statistically significant amount of microphthalmia. And we're also looking at, in the detox enzyme, there's still a little bit induced there, but it's obviously a huge improvement over the unfiltered runoff condition. So I'm showing here two papers where all of that research is summarized, and these are both published as open access, so they are freely available to anyone with internet access. So that summarizes the work with the zebrafish and also with the juvenile salmon and the invertebrates, looking at the effectiveness of that bioretention treatment. So I want to bring this back to the adult coho salmon. And we have now shown that stormwater runoff, with stormwater runoff exposure, we can create pre-spawn mortality, but we wanted to know now whether we could prevent it by using bioretention treatment. So what we did is we constructed our own portable bioretention system using just 55-gallon drums, a series of 4 of them. And we used Department of Ecology's protocols for a slotted underdrain system, topped that with a gravel drainage layer, and then again the workhorse of the bioretention system, which is the bioretention soil media, which in this case was 60% sand and 40% compost. We topped that with some mulch, and then you see in that bottom right picture our water distribution system. So following conditioning with clean water, we brought the system out to Grover's Creek. We helped the tribe collect the adult coho spawners that we would then use in our study. And we brought out runoff that was filtered through the bioretention system. And in the bottom right image, you see some of the filtered, treated runoff coming out of the bioretention system. So that is the third treatment that we added to this system. We have the adult salmon in the control well water. We have adult salmon in the untreated highway runoff, in this case, showing 100% symptomatic, leading directly to mortality. And then we see the third treatment is the treated runoff, what would happen. Now this complicated figure is just showing across 2 seasons when we collected runoff to do these experiments. So in 2013, there was a long dry spell at the beginning of the spawning run. but and the dotted line is cumulative rainfall. And the yellow bars are showing when we collected stormwater for our experiments. So we were able to get in 2 tests towards the end of the spawning season in 2013. And then in 2014, we were able to get stormwater runoff and fish concurrently in order to do an additional 3 tests. This table summarizes those results across those 2 years. You can see that in the first experiment we did in 2013, we did just a 4-hour exposure. And we saw there that all of the fish in the control water were alive. In the untreated runoff, we saw half of the fish were dead and half of them showed pre-spawn symptoms, pre-spawn mortality symptoms. And in the treated runoff, we saw all of the fish were alive and healthy looking. And we were concerned that the fish in the treated runoff would eventually show the same symptoms. It might just take a little bit longer. So we brought out our exposure period to 24 hours after that. And what we saw was consistent across the remainder of our experiments. We would take the fish out and look at them at 4 hours, but then at that 24-hour time point, all of the fish in the untreated runoff were dead. All of the fish in the control water and the treated runoff were alive and healthy. I have some videos here that show that. So in the well water, we see a regular control fish. It's active and responsive to stimulus. In the unfiltered stormwater runoff here, I'm showing 2 videos with varying severity of symptoms. The first one is just simply a little bit lethargic, and the second one is shows that loss of equilibrium. Now in contrast, here we have a fish exposed to the filtered stormwater runoff, and obviously very responsive to stimulus and active as well. So in summary, we've definitely seen that urban stormwater runoff is a very serious threat to aquatic ecosystems. We know that it can cause multiple symptoms of toxicity across different organisms. We see mortality and cardiovascular toxicity in developing fish. We see mortality and reproductive impairment in aquatic invertebrates. That's some of the material that's in that paper, but that I didn't show today. And we see mortality in both juvenile and adult coho salmon. And very importantly, we have seen that soil bioretention has the ability to prevent this acute toxicity. So the hope is that including mitigation strategies like low impact development and also source control, we hope that even though this is not a forest, that through those methods we can help it act more like a forest. Please feel free to contact me if you have any questions.